All right, so yeah, totally forgot about the quiz. My fault. Don't worry about it. So we'll just discuss this paper, and then I'm actually going to assign you the next paper a bit early, okay? The Halverson paper, because I want to submit questions to him so we can discuss it and have a chance for Dr. Halverson to get his answers back to us. But we'll talk about that in a little bit. In the meantime, let's talk about this paper. Who wants to start us out?
I'll head off the discussion if that's okay.
Yeah.
I had a question about the procedure. I think I understood most of it, but there is one part that they mentioned. So they're taking different steps before they realized what was going on, right? And they found the basidiomycetes, whatever. And this is what they said. 1 second, I'm trying to find it. So it said, we estimated transcript abundance by mapping raw reads back to a single pooled metatranscriptome assembly and binning by taxon. Mm-hmm. And I have no idea what that is saying.
Oh, okay.
Can you explain that maybe a little bit?
So, do you know what a metatranscriptome is?
Yes.
Okay. So we've got all of our transcripts just sequenced without regard for where they came from. So we take the lichen, we sequence all the transcripts that are happening. If we want to find out after the fact where they came from, then we can compare those little short metatranscriptome reads that we have against a big concatenated kind of reference.
So is that the binning? Is that what they mean by that when they say use the raw data and bind it? Whatever.
Yeah, and so they've got, 20 million of these short little reads. They're going to start clustering them together based on taxonomy. And so you match them up against this big reference, okay? And at that point, you can start making some calls about what species they may have come from.
Got it.
And so then we bin all the ones that came from an ascomycete versus a basidiomycete.
And that's why they could draw those not conclusive evidence that they were related, and hence they could do more research.
Yeah, let me see if I can find. It's figure 1C. Let me pull it up for you so we can all look at it.
Yeah, that'd be great.
Okay, so these little plots right here tell a pretty big part of that story, and what they're looking at is the transcript abundance. So they've already binned them, and now they're going to count them.
Okay.
How many of this transcript is in this particular taxonomic group versus that one? And after they've counted them, they're looking at log fold change. So twofold would mean you get double the amount of the baseline. And so this is a way of just kind of comparing if there's significantly more or less of a given transcript in a certain bin.
Okay.
So that's essentially what they're showing in these plots here, broken down by, well, they started with ascomycetes because that's what they were expecting, right? And here's the alga, and then when they allowed themselves to bin those transcripts, not just against the ascomycete database, but all the things, they saw that a bunch came out as basidiomycete.
Okay.
So I think that was the like smoking gun that something weird is going on. Was this plot? Yeah.
Perfect. Thank you so much.
Mm-hmm.
And I know I have another question, but I can't remember exactly what it was, so I'll look through it and I'll be back.
Okay.
Thank you.
I've got a question.
Yeah.
So when they talk about like let me pull up so that I get it right. So it's like the fish targeting.
Yes.
What exactly? I read through it and I didn't really understand what it was or how they did it. I guess.
Fish is a pretty cool technique. It's been around for a while, but it's still super useful. It's fluorescent in situ hybridization. And so, in essence, you're making like kind of like a little PCR primer that'll match up with a known section of DNA, but attached to that little probe is a molecule that when it's bound to DNA, it will fluoresce in a certain color.
So it's actually a molecule on the probe that they're sticking down into that ends up fluorescing.
Yeah, so it's like a little DNA probe, and it's going to match up with a known, it's the reverse complement of a known piece of DNA. When it does bind, that little molecule attached to the DNA probe will fluoresce. If it's not bound, it won't fluoresce. And so then you just shine a certain wavelength of light on there, and you can get back the fluorescent. And so this, it's really cool because it lets you can even add several fish probes at once with different colors. And so you could see where different cells are microscopically with that fluorescence.
Okay, thanks.
Mm-hmm. Let's see another hand.
Yeah. Sorry, I came in just about 4 minutes late. I was in a different. I thought we were going into the live classroom chat, but this isn't.
This is where everyone
Anyway.
was. I just followed.
Yeah. No, you're fine. I wanted to talk about the volcanic acid. Could you kind of go over that? I know it says that it was in part of the paper. It says that it causes the lichen to appear yellowish.
Mm-hmm.
Is that also what starts to break down the rock that it's on?
I actually don't know anything about that particular acid at all.
Well, they mentioned it quite a bit throughout the paper, so I was kind of wondering if that had a major significance to the paper. Was that what they were testing as well to kind of link these lichens together?
I just kind of glossed over whatever that acid was, and the way I read this was they used that acid production to tell these two species of lichens apart.
Okay.
So it could have been anything, but they had two species of lichens. One looks more yellow.
One looks more dark brown.
Yeah, and it makes this acid. And so that's how they tell them apart. A lot of times with lichens, you do have to do a little chemical test on them. So there's little reagents you could put on there, and if it fizzes, you got one species. The reason you have to do that is because these two quote-unquote species of lichen have identical genomes.
Oh, all right. That makes sense.
Yeah, so it's a conundrum in the world of lichens that these two lichens have identical genomes, but they look different from each other. And they do different stuff.
Okay. Thanks.
Mm-hmm.
Dr. Zahn, that's what I was going to ask. Actually, I'm piggybacking on that. So it said, so the difference between these ferns, I guess, is that, and they all have this like cystobasidium, I think is what it said. Is that it was like previously only known like on a couple places or something. So I guess I was just curious if that meant that every single fern species had cystobasidium on it and they just never somehow knew before.
You mean lichens instead of ferns?
Or lichens, sorry, yeah. Lichens.
Not every.
Oh, not every.
But many, yeah. And so they're saying that the actual difference between these quote-unquote two species of lichens is the presence of that Basidiomycete yeast. That's why some of them look yellow and are making this acid. So it's actually the same species of Ascomycete and the same photobiont, but you add in this third partner and it looks like a different organism.
Okay, cool.
And it does different things.
So maybe I was misinterpreting the paper, but it seems like they discovered this by accident.
Yeah.
Okay, so they did. So they were just comparing a couple different things, and they were like, "Oh, this is here."
Yeah, I mean, they even described kind of like the first experiments that they did that didn't make any sense. That's why they went with transcriptomics. Was they were trying to figure out why two organisms with the exact same genome could behave so differently. And so the obvious hypothesis was, well, one's just transcribing different stuff. Okay, so they've got the same genome, the same instructions, but one of them is just using a different set of instructions. And so that's why they went in here doing this transcriptomic analysis. Now, they did it with the fungus and the alga to see maybe one of those partners, we're not sure which one, but maybe one of them is just transcribing different things. I would ask if anyone thinks they understand what this plot is. 1A, 1B. Have any guesses? See some hands up. I don't know if those are for other questions, but.
Mine is up for a different question, but are these gene families?
It's just molecular clock, basically, based on the genes and relation, right?
Transcript and not genes, but
sorry, transcript.
Yeah,
yes.
yeah.
Yeah, but
And
it's basically looking at different fungi, I guess, and how they relate to track back where the differentiation happened, right? At least that's.
Yeah, and so we've got the two species of lichen shown here in these two different colors. Now, what you would expect if the difference between these two species of lichen was that they are just transcribing different stuff, you would expect all the same color to be kind of clumped together over here. What we see instead is that the colors are just interspersed kind of randomly for both the alga and the fungus. And so if B. tortuosa was transcribing a nice set of different stuff than B. fremontii, we would see them clustered together. So this is essentially a phylogeny, but instead of looking at the whole genome, we're looking at these single nucleotide polymorphisms and the numbers of the abundance of certain transcripts. So this panels A and B here is the result of the original experiment they thought they were going to be doing and publishing, essentially. And I bet that they thought all the colors would be clumped into basically two distinct branches on this phylogeny, and then that was going to be their paper, right? So you'd be able to say that the actual difference between these two species, even though they have the same genomes, essentially, is that they're transcribing things differently, but they did not find that. And so, like good scientists, they said, okay, well then what is going on? And they started digging deeper. All right, James, I think you had your hand up next.
Yeah, I hope this is a coherent something, but my understanding of a lot of what transcriptomics is it's measuring not just the transcripts, but also which transcripts are transcribed given specific stimuli or life stages. You see the transcriptome that you would see on a measurement of it differently at various stages, like an embryonic stage would be different than an adult stage typically. Was that anything to do with why they were using transcriptomics here, or was that basically just trying to discern the difference based on identical genomes?
Yeah, I think it's the latter. You're right that transcripts will change over time, like if you add salt or raise the temperature or look at a different life stage. Organisms are using different parts of the genomes at different times, and so transcriptomics gives us a really nice clear snapshot of right now what genes are being used in this organism. Not so much what genes are available, but which ones are being used exactly right now. And so you can manipulate things, like you can grow up some cells under one condition and then another condition and compare the transcripts. So they may have the same exact genome, they could be clones of each other, but they'll be using different stuff. And so in that way, transcriptomics do let us look at what genes are useful or being used in an organism under a given circumstance. In this case, though, it's just a nice alternative way of basically looking and seeing what's actually different between these two species, right? Why does this one look yellow and make the acid? Well, the hypothesis was, well, it's just transcribing different genes. And we didn't know why. And, you know, that was probably going to be the next test, right? Is why is it transcribing different genes? What condition has changed?
Okay, that makes sense. Thank you.
Mm-hmm. All right, who's next?
I can make a comment. I've had my hand up, but I don't know if I'm actually next.
Yeah, go for it. There's no rules.
Yeah, so, yes. So when reading the paper, like, of course, at the beginning, it talks about, like, what it's actually, like, made of, which, like, the ascomycete with the photosynthesizing center. And then they found that the basidiomycete was there, which was, like, the surprise factor or whatever.
Mm-hmm.
So I don't know if this is a dumb question or if it even makes sense. But, like, what would be the difference if they would have found, like, a zygomycete or something like that instead of a basidiomycete? Like, that could that even exist like that, or it doesn't really work?
I think it could. We probably would have found that before now, because the genomes are so different. But maybe not. I think what was aiding in us basically not being able to detect this before was that these are really tiny basidiomycetes, just like little yeasts.
Mm-hmm.
4 microns, and so they just blended in with everything when we were looking at these under a microscope before. They did mention that, you know, these lichens have been sequenced before, but nobody ever found basidiomycete sequences in there.
I guess when I was, like, looking at it, I was just thinking, I was like, well, if we never found basidiomycetes, like, what else could we potentially not be finding that could be attached to these things or other things, you know? And which made me think of, what about zygomycetes? But I don't know.
I think their methods are capable of picking those up. We've never looked at lichens this way before. But you can see here in panel D, they took all these transcripts and instead of blasting them trying to match them up against ascomycetes, they did asco, basidio, and then they found some others that didn't match, pictured here in black, right? So there do appear to be some other stuff in there. Those numbers are pretty few, and it's very possible that we just don't, that those are ascomycete and we just don't have them in the database that we're matching up against. But they could be something else weird. And so if you wanted to find a 4th partner, this would be the way to go about it.
Okay, cool.
Dr. Zahn, I'm not sure if this is a totally random or it is off topic, but I was kind of curious about figure 4. If you could kind of explain that, like between the differences of B and C and how that correlates to A. Just kind of confused on that whole figure.
Well, they included a video. I don't know if anybody found this.
I did not watch it.
All right, it's in their supplement. So this might help. So purple is Ascomycete hyphae. And we're zooming down on the top of this, and the green is the Basidiomycete yeasts. So as we kind of zoom through here with the fluorescent microscope and pick up everything, you can stack all those images together and reconstruct like a 3D analysis. And so they're showing that these Basidiomycete yeasts are embedded in the Ascomycete tissue. So that's pretty cool. So if we go back to the paper, maybe this figure will make a bit more sense now. This is showing that same thing. It's just a top-down view of a little cross-section of a hypha. The red is chlorophyll. It autofluoresces, so you throw UV light on chlorophyll and it just turns up red. Okay, so you don't even have to stain it. So that's our photosymbionts. The purple is Ascomycete, and then the green is Basidiomycete, little blobs, those yeasts. Okay, so that's the top-down view. This is a cross-section, basically looking sideways across the lichen. And there we can see that there's this photobiont layer, and then the Ascomycete layer in purple, and embedded in the top of that are all these little yeasts. Does that help make sense?
Yeah, that video helped a lot actually. Now,
all right, let's watch the second little movie also. In that case, so this is looking at a cross section in slices, and so they're showing essentially the same thing, and boom, there's that figure.
And then that's the other figure. Okay.
And then if we take off the side so we can see along the edge, that's the other part of that figure. So I think it's really great that they included this video.
Yeah, that makes a lot more sense.
Is that something cool that shows up? Is that just made when they do these tests, or did they hire somebody to do the graphics for this?
No, you can make that.
Okay.
Yeah, I'm guessing. I didn't look into the supplementary materials, but it was probably done in R with the Z-Stack package.
That's good to know.
Yeah. That's what it looks like to me anyway. Material acquisition, extraction, transcriptome sequencing, gene abundance, mapping, tree dating, sequencing, phylogenetic analysis. Okay. Developed a bunch of probes, tested them all. So a lot of work is going into this paper. They do their in situ hybridization. Okay, Zeiss axioscope, yep. Fluorescent. All right, so there's the software. So yeah, no need to hire anybody. Hayden.
So I don't know if you saw, Kate asked a question in the live stream chat because he wasn't gonna be able to be here today, and I had the same question. Basically, they mentioned that they were finding Basidiomycetes previously, but they just thought they were contaminants and were like just tossing them. Were they being found in like every single sample? Like, why were they just considering them to be contaminants?
Because they weren't being found in every example. So if you crush out the lichen and try to sequence it, you get back just a bunch of noise. Okay, that's because there's multiple partners, right? There's the photosymbiont and the what we thought was the only fungal partner. So you separate them out. You go in and pick them apart with tweezers, essentially, and sequence just the fungal part. And so that was usually done well below the photosymbiont layer, so that we were trying to mycology. Okay, so we take that and we just pull some out and sequence it. And so that right there means that you're probably not getting those basidiomycete yeasts straight from the get-go, because you're physically separating them without realizing it, just on accident. Now, when you do accidentally get some every now and then in your sequencing, it looks weird and you just throw it out because you're not expecting to find them. And so it's not what you were expecting, so you think your experiment failed, essentially. So that's, I think, what has been going on in the world of lichen sequencing until pretty recently.
Okay, thank you.
Mm-hmm. Christina.
Okay, so I found my question. Sorry, I was trying to remember what it was. So they mentioned right in the beginning when they're looking at they're basically finding some evidence that there's something else going on, and they mentioned then that they found that there was very specific genotypic DNA from Basidium mycota unique to each lichen. But then they only mentioned 2 species of, and I'm sorry if I butchered this, Basidia or something like that, I think, is the species. So were there more species identified, or are we talking about different parts of the genome from those 2 that were influencing?
More species. Yeah, more species have been identified since. So
there have been. Okay.
Yeah, so that's what these trees are about here.
Okay.
And essentially, they saw this and they're like, okay, well, that's weird. The picture's starting to come together. They're noticing these yeasts. And so the next part of their experiment was to go out and collect lichens from all around the world.
Okay.
And they mentioned they got lichens from 6 continents, right? And they looked for these. And they found them in a bunch. And so here are 8 clades of lichens where they found these Cyphobasidiales fungi, right? And they're kind of spread all out. This tree, this tree right here is basically kind of zoomed in on these 2.
I see, but there are other ones that they have identified. They just don't mention those.
Yeah, so they found the initial one was right here in Bryoria. Okay, so that's showing up right there, and then over here they found some more. Okay, so they've got, and it does seem to be that each little species of this yeast apparently is at least somewhat specific to certain lichens, and that's across the whole range of the planet, essentially.
Great, thank you for that clarification. I assumed that was the case, but just wanted to clarify.
Yeah. Dalton.
Okay, I got a few questions. But so the first one would be, did they just make up the name for a new order in this paper?
No.
Okay.
No, I don't think so.
There is, on the bottom right there of this, it says, using current classification criteria, the lichen associated, as in the new order, Cyphobacillides. Did I just interpret that wrong?
Oh, yeah. It looks like they did make that up. Let's see, 19. Okay, so they didn't do that in this paper. They did it in a previous paper, and they're now citing that. So these are the same authors, Spribille, and in this paper they're describing this new order, and up here they're citing it.
So that's pretty big. What kind of like verification process does just naming out a new order go through?
A big one. Let's look this up. Ah. New lichen inhabiting lineage in the Cystobasidium mycetes. All right, so here's the paper. We do a bunch of taxon sampling. We find a ton of these things. We deposit all their sequence genes in NCBI. We say where we found them, do morphological and molecular analyses. We design new primers for this group, amplify their genes, and build a phylogeny. Let's see if we can find. There it is. Okay, so yeah, a lot of work goes into coming up with a new order.
So has that been accepted by now? I don't know how old this paper was. Okay.
Yeah, this is 5 years old here, so this tree may be modified since then, but I doubt that anybody's going to be able to disprove that Cyphobasidium is monophyletic or in the Cystobasidium mycetes. Yeah. All right, you said you had more questions.
Yeah, I can keep going if nobody else has.
Yeah, go for it.
Okay. The next thing would be, did you already explain what makes the thalli hard to grow in lab? Because they made a point of saying that it was pretty hard to do that.
It varies from species to species. Some are easier than others. I'm guessing this one just, it needs certain conditions that we don't quite understand well enough to grow them in the lab. That's common with a lot of fungi. You can make new media to try to get things to grow, but it's kind of shooting in the dark, and you're just trying stuff to see if it'll grow, and if they're slow growing, that's a problem. I found one, I think is a new species of fungus growing in the Great Salt Lake on a piece of floating wood. It's been growing for 3 years now, and it's about a centimeter across in my petri dish. So that's ridiculous. And it's growing on a weird like saltwater wood media that I developed also. So maybe there's a way to get it to grow faster, but at least it's growing.
All right. The next thing would be, so they talked about something rather specific where they used electron microscopy, right?
Mm-hmm.
And they said that it wasn't finding, essentially, it wasn't detecting the Cyphobasidium due to it being embedded in polysaccharides, which that's just like complex sugars, right? Polysaccharides. Yeah. So why would being in sugar affect an electron microscope to where it wouldn't be able to see them?
If you're coated in that sugar and you're doing the scanning electron microscopy like this, it just looks like a lump, essentially. And so scanning electron just gives you the surface. And so if you're looking at the surface of something and you see some lumps that don't look like distinct cells because they're encased in this glassy sugar stuff, then yeah, it's pretty reasonable that you wouldn't think, oh, that's a different cell.
Okay, so they would have needed maybe more of a side view instead of a top-down view.
A side view and maybe transmission electron microscopy, which is a huge pain in the butt to do, but yeah.
Okay.
Transmission, the TEM, it involves like a really, really sharp knife, and you embed your sample basically in a polymer and then slice off layers that are so thin that electrons can actually pass through them. And that way you can kind of like see through the cell in layers. Notoriously difficult to pull off and can take years to get like good pictures. So it's not surprising that nobody's done that on
It's just not worth the effort.
Yeah, like who cares? Like, what are you gonna find? Some surprise basidiomycete yeast? I don't think so. Yeah.
All right, and then the last question I have here is, oh crap, I deleted half of it. But it was talking about the multi-template PCR bias. Can you explain what the bias is?
Yeah. Let me pull up a resource. Okay. Typically, when we're using PCR to do some sequencing, you know, we add in our primers, and the most common barcode for fungus is ITS1 and a lot ITS2 also, but this is a good marker. And so we use primers like this forward primer, that reverse primer, and we sequence it both directions, merge those two sequences together so we get like a nice read that we believe in, because it would sequence the same base pairs both directions. So we believe that, and we get ITS1. Okay. This is going to be like 350 bases long-ish in most fungi, but it varies. In bacteria, there's regions like this also that we use to sequence for species identification, but they're like a set length. In fungi, the length can vary. In some zygomycetes, the ITS1 region is like 2,000 bases long. Okay, so we have a problem. When we do PCR with these primers, if this region is actually 1,000 bases long, then you're amplifying this giant chunk, and the PCR protocol you use might not be set up to amplify that long of a chunk of DNA. That makes sense so far?
Yes.
All right, so we know like there's the annealing temperature, then the extending temperature, and then we melt again, anneal, extend. If you don't leave it in that extend temperature for like 3 minutes, then it's not going to have enough time to amplify that big chunk. What they were talking about, where was it? I think they found like some of these had a 500 base pair insert. Where was that in the paper?
I would Control F and just type in, they spelled out PCR, like the polymerase chain reaction.
There it is. Okay. 595 base pair group 1 intron insertion downstream of the primer binding site. Okay, so ITS1F, there's that primer. So ITS1F, and then there's almost 600 base pairs extra that we weren't expecting. And so that's going to be amplified at a lower rate than the shorter sequences.
So they weren't leaving it, they weren't letting it sit for long enough, essentially.
Yeah, and even then, PCR does have a bias towards smaller reads being amplified. Okay, so even if you leave it for 3 minutes at the annealing temperature during every cycle, the short stuff's going to amplify faster. And if you're doubling with each round of PCR, just having a few extras at the beginning, if you're doubling over time, you wind up with millions and millions more of the short reads than you do the long ones. And so the long ones can get swamped out when you go to sequence afterwards.
All right, that makes sense. Thanks.
Yeah. Leah.
This might be going back to the beginning a little bit, but my internet is being spotty. I was just wondering how exactly the yeast like affect the phenotype. Like, what about it makes the lichen change colors?
No idea.
Sweet.
Yeah, they didn't make it that far. They did like 4 papers worth of experiments already. I think they probably just found a stopping point. I would not be surprised if they're working on that very question right now as a separate paper.
Okay, cool.
Yeah. Can you guys hear that? My neighbor's like busted out the leaf blower for the season. Driving me mad. All right, anyone else?
Yeah.
Questions, comments about this study?
I had one. So
Okay.
on page 490, on the bottom right, it says that the internal cellular structure is not uniform across lichens, and the composition of extracellular polysaccharides is poorly known.
Mm-hmm.
Why is that? I guess, like, to me, as an inexperienced scientist, I don't understand like why that would be such a difficult thing to understand the composition of it.
Just a matter of resources, right? I mean, how many lichen species have gotten this kind of treatment? Just a few at this point. So it's just a matter of going in and doing it for all of them if you actually care to know. What they're talking about here is not all lichens have that nice defined, like, here is the medulla, here is the photobiont, right? And then here's the surface of the lichen. Sometimes it's more complex. There's like a couple layers of photobionts. Sometimes there's photobionts kind of embedded deeper into the medulla rather than just a nice little layer. And so not so much cellular, like intercellular structure, but or intracellular, but that each lichen kind of has its own body shape in essence. And so this, we may not be able to see this nice little pattern like that in each lichen.
And like, do scientists just not think that it's important to know, or if there's just like not enough importance within the ecosystem between the algae and the fungi, or is
Scientists,
it just is it like a funding?
Yes, it's a funding thing. I guarantee there's 100 people on this planet who would drop everything they had to go out and do this for every lichen on Earth, but it's expensive. And so, and it takes time. And so they'd have to write these grants and get them funded in order to do that. I mean, they're naturally curious people, otherwise they wouldn't be geeking out over lichens like this. So yeah, people want to do that. It's just a matter of whether or not we can have the time and energy and money to get it done.
And that's science for you.
Yeah. All right, I'm checking out the chat here. Sydney says, feels like most papers give a clear explanation of what's next, but this one doesn't really. I think we just kind of talked about that. Like, let's see how widespread this is and see if they follow the same sort of pattern of being embedded within kind of the surface of the ascomycetes in a bunch of different lichens. And then an obvious what's next for me is, what is it that these basidiomycete yeasts are actually doing. Why, if you have these, do you get a different phenotype? I mean, they even found them in the brown version, the Fremontii, right? They're there, just not nearly in the same abundance. So what is it about reaching some critical threshold of having these in your tissues changes you phenotypically so dramatically? That's the next I would probably shoot for, and I bet they're already doing it. Okay. All right. If there's no more questions, I'm going to go ahead and assign you kind of like one class period early, this Halverson et al. Paper. Okay, so here's the paper. This is starting to get at some of those ecosystem services of fungi, like how they're breaking down stuff in nature. And then there's a nice little podcast, just about 15 minutes long, with the author talking about this particular study. This is one of those author manuscripts that I got from the author, and so all the figures are annoyingly at the end of the paper, but they are in there. So look for them down at the bottom. What I want you to do is to come up with questions for the author. We were going to have him join us, but I got the dates mixed up because I suck at life right now. And I told him to join us today to talk about his paper, and then it occurred to me, we haven't actually read your paper yet. Sorry, Hal. So he can't make it next week, so we're going to send him some questions on Tuesday, and he'll answer them for us in either like a little video or just written out. And so we'll have those to work with when we discuss the paper on Thursday. Everyone clear on that? I'm going to stick those instructions online.
So basically send the questions we have by Tuesday when we have class.
Yeah.
Okay.
So on Tuesday, I'll have you just upload your questions into the chat box or whatever, and I'll send those off.
Okay.
Assuming that the paper and the video clip are just, sorry, they coincide with each other, right?
Yeah, it's actually just like a little audio clip, but it's one of the editors asking the lead author, Hal Halverson, some stuff about the study. So that'll kind of help you get an idea of the sorts of questions that an editor would ask somebody about their paper.
Okay.
So Dr. Zahn, is there also going to be a quiz, or are we doing those questions instead of the quiz, or what?
Yeah, these questions are going to be in lieu of the quiz, so make them good, you know?
Yeah.
And give me more than one if you can. They don't have to be about the technical details of the paper either, right? They can be, you know, like, what was the most surprising thing that you found, or what was your favorite part of the study, that kind of stuff. But if you're going to submit one of those questions, also give me a good question about the actual paper, the interpretation of the results, or like something to clarify some of the methods. So some of the same sorts of questions that we ask about the papers that we've read in here, but also a chance to actually talk to the author and kind of see what doing this research was like.
So what kind of favors are you doing for all these authors that you're able to get them on? Are you just friends with them?
Yeah, I'm just friends with Hal Halverson. we did grad school together at the same time. So
Thank you
I
for sending
remember
that to
him
us. That
starting
was
to work on
cool.
this. What's that?
Oh, I'm so sorry. I was just saying thank you for setting that up for us. It's really cool.
I'm sorry that I screwed it up and I got the dates wrong, because he was gonna like video chat in with us, which would have been.
That would have been really cool.
cooler.
But even just having him respond to our questions, that'd be
Yeah.
awesome. So thank you.
Well, thank Hal when we get a chance.
That too.
Because it actually is a really cool paper. He's not a mycologist. He is a freshwater ecologist and mostly works with like algae and insects decomposing leaf litter and stuff. But this paper happens to be about fungus. So I think it's a kind of neat way of bringing a paper about fungi into our class where we can see how they actually interact with other organisms and bigger biogeochemical processes, because that's kind of what we're going to be talking about next week, is how fungi drive biogeochemical processes. And so this is just a neat example of that kind of in practice. All right, anything else?
I just want to make sure that I heard this right, that we need the questions by Tuesday, not Thursday like we normally would.
Tuesday, not Thursday. Yeah.
Okay.
And those questions will be graded on how much effort I think you put into it. So if you ask like a really stupid question that's answered in the paper very clearly, like if you said, on what days did you collect leaf discs? I'm not going to give you very many points for that because it's right. So put some thought into it. Read the paper first. Think about it for a little bit. Listen to that podcast and kind of see the sorts of things they talk about, and then come up with questions that you have just about the work, about what it's like to do that kind of work, what kind of methods go into it, all that stuff. So it does not have to be a deeply technical question about one of the methods. You don't have to understand everything. Hal is aware that you are undergraduates reading this paper for the first time, and he's a nice guy. He's not going to like make fun of your questions. But also he's going to have really interesting insight for you as like an early career scientist about what doing this sort of research was actually like. Behind every paper, there are actual human beings. It's easy to forget about that.
Sorry, did you say how many questions we needed to come up with? Or just come up with a bunch that we think are worthy?
As many as you want to ask.
Okay.
At least two.
Okay.
So maybe one about like interpreting the results or a method you didn't understand, and maybe one that's more general about kind of like what's the big picture, you know, what's the next step, or about what it was like to do this. You know, if I wanted to do a study like this, what kind of skills would I need to learn, that kind of thing.
Okay, thanks.
Mm-hmm. All right. And with that, I will leave you to it. So read that paper over the weekend, come up with some questions, and I will see you next time.